You've probably heard that this meeting is recorded. Good morning, afternoon again, everybody. Today's a special day, especially for Jake here, because this is his thesis defense. I'd like to welcome everybody in this Zoom meeting, and I'd like to especially welcome the members of Jake's committee. Dr. Mike Chintako is here. Also, Dr. Jonathan Awaya. They're all with us here today for Jake's thesis defense. So before we have Jake take over the screen, I would like to introduce him very briefly, and then I'll let him take over. Jake has been a student of mine since 2010 in the College of Agriculture, Forestry, and Natural Resource Management, and he finished his undergrad degree with focus on tropical horticulture. Was it, Jake? Tropical horticulture?
General agriculture.
General agriculture.
It's not here anymore, but.
Yeah, general agriculture. I took several of my classes, and that's probably how he admitted. He was fascinated with composting, earthworms, and everything that is dirty. He admits that he likes water. He's a surfer. He's from Maui. He went to Luna High School, and I believe when he finished college, he went back to Maui and worked there as a farm manager. He came back to school for his master's degree in 2007. Continued to work at a full-time at our college, Hilo, while going to school, performing his research and taking his courses for the master's degree. So he now resides in Hilo with his wife and son. And without much further ado, I'd like now to give the screen to Jake. Jake, take it away. Thank you.
Okay. Thank you, Norman, for that lovely introduction. Well, I'm Jake for all of those that are out there that I don't know personally. Well, let's jump into the effects of vermicomposting and pre-composting on fecal coliform bacteria occurrence in animal manures. So as a brief outline here for my slides presentation, I'm going to go into the introduction of how I got into this particular research, some of the problems behind it, and then the methods on how I conducted my research, the results from my research, and some implications from my results, as well as some future research that could improve upon this particular area of focus. And then also, I'm going to wrap it all back together with a conclusion and the main take-home points from this research. So I'm a very food-oriented person. There's a saying out there that there are those that eat to live, and then there are those that live to eat. Well, I am the later of the two. I enjoy eating, and outside of consuming food, I also have a passion for the production of food, and in particular, sustainable and regenerative practices for this. And this was fostered by growing up in the islands and seeing how we import roughly 90% of our food into the state of Hawaii. And this is a huge concern, especially if you live here and you have want to make sure that there's food security for our state. And so there's all these resources out there that could be utilized to produce food here as well as globally that are just being wasted or not utilized properly. One in particular, one resource is manure. Now, manure use in agriculture can be traced back to the Neolithic era, which is roughly 12,000 years ago. There's a study by Bogard et al. 2013 that used carbon isotopes to see that there was these groups of people back then that used manure in increasing the growth and productivity of their cereal and pulse crops. Now, if you fast forward to today, we still utilize these manures in our agricultural system. We just have a lot more concentrated amounts of these manures available to us. And especially with the large amount of people that are on this planet right now. There's all sorts of different types of manure resources out there, from chicken manure to horse manure to turkey manure to guinea fowls. If an animal poops, someone's potentially used it in growing food. Now, in modern times, we have a lot more mechanization than what was had during the Neolithic era. So instead of leaving our animals in one particular area and then growing our food once we've moved the animals in that particular area, we have machines that we can use to spread manures via tractors, or expel the liquids from the solids and use the liquids in irrigation lines on our fields. Manure is a different form of fertilizer for plants than what we're used to, which is chemical fertilizers. It also contains a similar nutrient component that other fertilizers have. So it contains nitrogen, potassium, and phosphorus, which increase the yields and productivity of plants, especially crop plants. And some manures have more of this nutrition than others. For instance, chicken manure has higher concentrations of nitrogen, and then bat guano, on the other hand, has higher concentrations of phosphorus. So there's all sorts of these different manures that can increase our production for food. And it also, since it is an organic material, it increases organic matter content in the soil when applied. Now, this is a graph from the FAO. And on the y-axis, you can see this is the annual application of nitrogen in megatons to agricultural soils. And then the x-axis is 6 decades. So from 1961 to 2017, this is the application of nitrogen per megatons annually. So you can see here that there is a consistent increase across the board from manure applied, or left on pasture, manure applied to the soil, and then also chemical fertilizers. Now, with the invention of the Haber-Bosch method, which pulls atmospheric nitrogen out of the air, the use of manure is kind of dropped down significantly, since it's a little easier to use pelleted fertilizer versus calculating your nutrition via manure. And also your means of spreading is a lot easier. That is a potential why the increase of chemical fertilizers in this chart is significantly higher than manures. Another big reason is that the use of chemical fertilizers, it is a sterile product. There is no biology to the chemical fertilizers, whereas animal manures contain microorganisms, and it is alive. And so this has the potential to cause harmful human pathogens, or contain harmful human pathogens like Listeria monocytogenes, Salmonella species, Escherichia coli or E. coli, as well as other fecal coliforms that can cause harm. Now, in particular for my research, I was looking at Escherichia coli. Now, E. coli in itself is a gram-negative, facultatively anaerobic, rod-shaped bacteria which possesses both fermentative and respiratory metabolism. They're roughly 1.1 to 1.5 micrometers wide by 2 to 6 micrometers long. And then they can also be motile or non-motile, which means they can move or just adhere straight to a substrate. Now this is important because that means it is very adaptable to many different environments and many different substrates. That being said, E. coli is very persistent in livestock manure. It can be found in pretty much every animal manures out there, from cows to deer and other game and livestock, wild animals. It also, in our modern agriculture, we have confined feeding operations, which usually have manure stocking yards. These manure stocking yards, they sit in piles, and these piles can contain E. coli that can last for up to 4 months in these particular piles if the piles are not turned or managed accordingly. And in particular, there's a group of E. coli in the enterohemorrhagic family, or a portion of the Enterobacteriaceae, which is the family for E. coli, that produce Shiga toxins. Now, Shiga toxins are a toxin that produces hemorrhaging, and one condition that can be caused when an E. coli is consumed in the human body is hemorrhagic colitis, which is bloody diarrhea, and it can be a problem that can turn into another syndrome that is called hemolytic uremic syndrome, which 5 to 10% of people that contract it, it is fatal for. Outside of the harm that it causes, one positive note for it is that it is an indicator species for other harmful human pathogens, as well as a fecal indicator bacteria. There's a criteria for determining this that was developed by a man named Bonds in 1966, and he developed ideal traits or qualities of a particular organism to be an indicator species for other things. And E. coli fits the bill for these particular traits to be an indicator species. Now, with this is a graph here of the CDC showing the incidence per 100,000 people in the US. And these are all different serotypes of E. coli that produce Shiga toxins. And you can see here that the orange line, if you can see my cursor, this orange line is the total overall incidence of Shiga toxin producing E. coli. And this particular line is just, it's steadily increasing in the incidences of E. coli in our food system. Now, E. coli is very easy to move through our food system via manures, as well as through our environment, through like the rivers, as well as it can also be secondarily caught in the packing houses, or it can be due to contamination in a packing house into our food systems. And this is pretty prevalent because with a higher amount of these manures sitting around in stocking yards, there's a higher chance for contamination in our food system via these different mechanisms I just expressed. One particular bill that came out in 2011 in the Obama era was the Farm Bill. And in this Farm Bill, there was a particular subgrouping of it called FSMA, or the Food and Safety Modernization Act. And in this particular bill, it talks about having restrictions for the use of untreated manures in crop production. So it makes the use of manures a little more difficult to be utilized. Now, with that being said, the United States alone has 26,000, roughly 26,000 confined feeding operations just for cattle alone, making up roughly 31 million head of cattle in just the US. And in this particular picture here, this is a manure yard in Nebraska, or a feed operation in Nebraska. And in the background, you can see, if you can see my cursor here, this is a mountain of manure in the background. This is not a geological mountain, the one closest to the foreground here. And like I was saying, E. coli can survive for up to 4 months in a pile that is not turned or managed properly. So this is just one feeding operation. So this is a large amount of manure that comes out of these operations. And this is just cattle alone. This is another feed yard in Texas. This facility expresses the solids from the liquids in their feeding operation. And the center image here, that is a manure lagoon. So the liquids of the manure lagoon can be utilized on non-crop production, something that's not going into the food system. So like biofuels or food fiber or something along the lines of that. But these manure lagoons often get underutilized and are just sitting there being a large petri dish for harmful human pathogens. To bring this closer to home, the dairy we had in Ocala, which is no longer running or operational right now, they utilized manure lagoons as well. And during Hurricane Lane, this lagoon overflowed into the nearby gulch and went down into our estuaries and down our coastline. So it caused not just like an algae bloom with the nitrogen that it had inside of it, but also just released these harmful pathogens that were subsiding inside these lagoons into our environment. Now, the Food and Safety Modernization Act is a positive as well as a negative, that it makes it more difficult to get manure used on crops that are going into food production, which will decrease the amount of E. coli that we could potentially consume. But it also prevents us from using this large amount of manure that we have sitting around. So for the to FSMA today, they have not come up with they have not determined a safe time for if untreated manures could be utilized or applied to a field before crops are produced. So they allow for the treatment of manures, which on here, this is a chart of their standards for microbial, the microbial standards for the manure, or biological soil amendments of animal origin. So anything that could be utilized as a fertilizer from animals. And in my research, like I said, I'm not focusing on Listeria monocytogenes or Salmonella species due to the fact that there was a higher cost to determine these two tests, as well as FSMA recognizes E. coli as a indicator species. So in my test, I'm looking at E. coli and fecal coliforms. So you can see here, E. coli, in particular 0157H7, which is a E. coli found in all animal manures. Has to be a non-detected using a method that can detect 0.3 most probable number per gram, as well as fecal coliforms have to be less than 1,000 most probable number per gram. Now, one thing on the FSMA bill they stated was the use of heat treatment, and in particular, they didn't state anything outside of heat treatment other than it has to be a method that can be scientifically proven to meet those particular standards. So one particular heat treatment method that is very well known is thermophilic composting. Thermophilic composting is a mechanism that utilizes thermophilic bacteria and fungi, which means they love high heat temperatures. So thermophilic composting is between a range of 39 to 77 degrees Celsius, or 102 degrees Fahrenheit to 170 degrees Fahrenheit. It is an anaerobic aerobic process, which means that it requires oxygen for the beneficial organisms to survive and thrive, as well as in increasing the air inside the piles. It requires usually large machinery and or heavy machinery to incorporate this air inside the piles. And you can see here that I have these two pictures on the left. The top is a windrow turner that mixes the pile as it drives over it. The bottom picture is a bucket loader that does pretty much the same mechanism as turning the pile that the windrow turner has. Now, both of these machines are upwards of $100,000. So these are expensive machines. So it requires a large capital to produce compost in these particular facilities, which would take in a large amount of manure. But thermophilic composting, the finished product improves soil physical properties as well as increases plant production via organic matter content, increasing cation exchange rate, also increasing infiltration rate in the soils. Now, thermophilic composting has many articles and research done on its ability to suppress plant pathogens as well as harmful human pathogens. Since the temperature of the pile can be to these high temperatures, it pretty much sterilizes the material inside, as well as it allows for other microorganisms to create competition. And as I was saying, the cost of running these operations and to start up these operations are relatively high, and it requires a lot of time and input. This is a facility I got to visit in Florida. This is a 15-acre plot that took municipal solid waste from nearby cities, and it utilized 7 bucket loaders to do this job. So is definitely a cost-intensive production method. Another method, like I was saying, FSMA didn't emphasize anything other than heat treating manures for making it biologically safe. So I wanted to look at another option, which was vermicomposting, knowing that it is not a thermophilic process. It is a mesophilic process, which means that it goes in between a temperature range of 15 degrees Celsius to 27 degrees Celsius, or 59 degrees Fahrenheit to 80 degrees Fahrenheit. It utilizes epigeic worm species. One species in particular that's well known is Eisenia fetida, or the red wiggler. And you're feeding these materials to the worms, and they're breaking it down because epigeic worms are a good surface decomposer. Now, vermicompost also achieves a similar result to thermophilic composting, where it improves soil physical properties as well as increases plant growth and production, with 2 added benefits: that since the material goes through the worm's alimentary canal, it physically changes the property of the material, which increases water, which can increase the water holding capacity of this material. So when you apply it, it also increases the water holding capacity in your soils. Another big one is that vermicompost has plant growth hormones in it. So when it's applied to the soil and to plants, it can increase the growth without making the plant look so succulent and attractive to pests that would attack other plants that have higher amounts of these higher amounts of nutrition to it. With some of my literature review and in researching this particular topic of vermicomposting, removing harmful pathogens, all the research articles that I could find were mostly and heavily oriented towards vermicomposting being a secondary process, and meaning that they altered whatever product they're using or material they're using prior to giving it to the vermicomposting operation. And as well as if there was a method that could remove E. coli, it wasn't the main focus of the research. The research was to look at one particular mechanism, not so much the end goal at being pathogen free for use in crop production. And so also, this is a facility that I got to visit in Florida as well. This is a very rudimentary setup, which required very low capital to start up. And that's one thing with vermicomposting that is so effective, is that you do not need high initial capital to start up these operations. As you can see here, this is just clear plastic over a hoop house, and they're using wooden boxes for holding their red worms. This particular facility utilized dairy manure from a nearby feeding operation, and they got the solids that were separated from the liquids, and they pre-composted this before applying to their worms. And so my particular research, I wanted to look at to see if, to determine the efficacy of vermicompost to remove E. coli and total coliforms from these particular animal manures. And then also as well as to determine if pre-composting is a necessary step to remove E. coli and total coliforms prior to vermicomposting. And so I utilized horse and hog manure, which the university farm has vast amounts of at the moment. We have a equestrian program, so I utilized horse manure from that, and they have a swine production program, so I utilized the manure from that. For my treatments and replications, the following slide, I'll talk to you about that. It's easier to look at versus me trying to describe. And then I will talk about my pre-composting guidelines that I followed. So I separated my two manures into two groups, and I have my treatment methods, or my treatments, which are pre-composted, which I have my standards in the next slide I'll talk about, but pre-composted the horse manure and the pig manure or hog manure, and then subsequently added it to my vermicomposting setup with 10 grams of Escherichia fetida, or the red wigglers. And I let that sit for 90 days, where my vermicomposted treatment was directly from the animal's rear end straight into my bins with Escherichia fetida for 90 days. I followed a national organic program, or the NOP standards for compost, which requires the compost to stay at a minimum of 131 degrees Fahrenheit or 55 degrees Celsius for 15 days, with at least 5 turns to have uniformity, so there was no anoxic zones that were not exposed to the heat. Just to note on here, before I went to this very basic method of turning, I tried to be lazy, and I utilized a system that I found from a university in Nebraska that used trash cans to force air into the material to cause it to heat up to create compost. Safe to say, me being lazy cost me a semester trying this particular method, And then you think I learned my lesson, but I thought increasing the size of the containers would generate more heat, so I didn't have to turn these piles. Again, fruitless. I wasted another semester. So I went back to turning these particular piles by hand. Now, compost piles have to be at least a minimum of 1 meter cubed to generate a significant amount of heat, as well as to insulate itself to keep that heat sustained, as well as you want to have an ideal carbon to nitrogen ratio of about 30 carbon to 1 nitrogen. So with my hog manure, I mixed by volume equally manure and wood chips so that I can get roughly 30 to 1 carbon to nitrogen ratio. This middle picture is of the turning process. The facility at the university is gravity oriented, so I didn't have to use as much energy to turn these piles. And then my picture on the far right here is my horse manure, which is already ideally at a 30 to 1 carbon to nitrogen ratio. Now, for sampling, I took samples from 3 different processes or 3 different portions of this method, where I took my baseline samples, samples after the horse manure and the hog manure were pre-composted, as well as after the 90 days of vermicomposting. To mention also, my baseline samples I took to determine the amount of E. coli that I had inside and total coliforms that I had inside my manures, as well as I took multiple samples and diluted it to determine which is the best dilution to have best readings on the particular method of enumeration. Now, I harvested from several different locations on the piles in each different treatment, so I could have a uniform sample that would express the whole container or the whole pile that it was in for its representation or the occurrence of these harmful pathogens. And so I weighed out 10 grams. I added that to 0.15 mole sodium chloride, and I shook that for 45 minutes and let that set for 10 after shaking for 45, so that I could pull off my specific aliquots to be turned into the analytical lab. I followed a procedure by Luis and Carmen for soil sediment pathogen enumeration, as well as Myers et al. 2014 from USGS to pull out my specific aliquots for sampling. When I sent them into the analytical lab, they utilized a method of enumeration called Colilert Quanti tray, which I'll talk about in some of the following slides. And then the data that I received back, we analyzed using SAS to look at using a proc GLM to look at the potential removal of E. coli or the occurrence of E. coli compared to baseline and among the different treatments, as well as, or among the different samples, as well as a t-test to determine if there is any significant difference between pre-composting prior to vermicomposting and direct vermicomposting. Excuse me. So here's some pictures of the soil sediment preparation for enumeration. Everything that you do when you do pathogen enumeration has to be very sterile, so there's no cross-contamination. And so these are the NaCl being autoclaved. These are the rotary shaker tables that we used. That was at 100 RPMs, revolutions per minute. Now, the Colilert Quanti tray, which is what the analytical lab uses to enumerate E. coli and fecal coliforms, is a easy to use method for enumeration. It's a you open the packet, you pour your sample in, you seal the packet, and you incubate it, and then you can read it under black light. Now the chemicals inside the Colilert Quanti tray are ONPG, which interacts with fecal coliforms and it turns them yellow after incubation, and MUG or MUG, which interacts with E. coli to turn them fluorescent after incubation if it is present in your samples. This particular method uses most probable number, which is an EPA recommended pathogen enumeration, versus colony forming unit or CFU, which could be one organism that cultures one particular tray, where MPN methods can be a better representation due to having multiple organisms for a better representation of the multiple organisms that are in the material that you're sampling. So here's on the left is the Colilert Quanti tray trays out of the incubator, and on the right is a blacklight reader. So here's one of the trays underneath the black light to see. The cell in the top is yellow, which indicates that it is fecal coliforms. But since it is not fluorescent, means it is not E. coli present in this particular cell. So these other fluorescent ones are having an interaction with some other organism or or another reaction. So with that being said, here are some of the results of my baseline and pre-composted samples of my horse and hog manure. So you can see here I have high levels of E. coli as well as total coliforms in both horse and hog manure. In particular, though, you can see my horse manure, which started off as 310 mpm per 100 milliliter. In my pre-composting step, it removed the occurrence of E. coli representation. So, which is very different than my hog manure, which actually increased during the pre-composted process of my test here. And this is potentially due to the fact that I didn't utilize a curing time for my compost, which is where the compost pile drops the amount of temperature that it has, and it allows for more organisms to come in, which creates more competition for the harmful pathogens. And so since I didn't utilize that, that is a potential reason for that occurring. Looking at my overall samples here, we concentrated them all into means. So on the y-axis, this is mean most probable number of all the samples that I have taken from the different treatments. And this is particularly for E. coli. And with this, you can see hog and horse are my baseline samples. C stands for composting, VC stands for vermicomposting, and C plus VC stands for composting plus vermicomposting. And as you can see here, any of the manure that went through a vermicomposting process has rendered the occurrence of E. coli undetectable. And this was a significant, this had significance to it with a 0.001 significance interval, as well as the similarities with total coliform occurrence. Same thing here. You can see that the manures that went through any of the vermicomposting processes made the occurrence of these total coliforms undetectable. And this was also a significant result in our statistical tests. This is the two graphs together so that you can kind of see the same form and shape that it has, that anytime I went into a vermicomposting process, it dropped significantly. And now this is important to note that for looking at this, the vermicomposting process it has shown to that it can remove E. coli from these particular manures that had a higher concentration of it at the beginning, as well as the need for this pre-composted step was not necessary for achieving non-detectable levels of these particular pathogens. Now, this is important for meeting the FSMA standards. So in this particular chart, I have adjusted my MPN to gram dry weight so that I can easily relate to the FSMA bills. And you can see here that the requirements for to be FSMA compliant for E. coli was not detected with a method that can detect 0.3 mpm per gram dry weight. So you can see here I'm well underneath 0.3 mpm, meaning that my samples have met the qualification for microbial standards for biological soil amendments of animal origin, as well as for total coliforms. You can see as well that it was less than 1,000 mpm per gram dry weight of fecal coliforms, and we're well below that one for fecal coliforms. And this has many implications because majority of the farmers have to follow FSMA compliance. If they do not meet these standards in use on their fields, they can lose their ability to grow produce and sell it to markets, as well as they could be fined if they continue to do this. So this is an integral part of manure stabilization and utilization for agricultural production. And with that, with our world population being above 7 billion right now, that means we have to increase our food production to meet the amount of people that we have on this planet, as well as when we meet this production level for consumption, we also have the same waste stream that will be generated by the production of all these foods. And that being said, there was a study by Schober and McGuire 2018 that looked at the amount of livestock production in developing countries as well as developed countries. And as you can see here, it is a steady increase for all animal production in these particular charts. So, and in particular, chickens in developing countries, which is the bottom graph, the black dots. This will pose a huge problem because these livestock production methods are usually confined operations, which means that there is these stocking yards of manure that are out there potentially causing harm to the environment and also not being utilized in a way that could be beneficial to the production of food. With that being said, my particular research didn't go into the utilization or the use of different other types of manures like chickens or cows or goats. And so this is an area that can be further researched to see if vermicomposting has the ability to remove harmful pathogens from these manures sources, as well as my particular research didn't look at using different epigeic worm species, which there is a need for that due to different microbial or different climatic regions globally. So there's a need to look at these different worm species to see if they have the same ability to stabilize manure to make it pathogen safe for food consumption. As well as here's an infograph from the World Bank showing the amount of the pounds of municipal solid waste generated per capita daily. As you can see here, the dark green dots dark green areas, for instance, this is upwards of 4 pounds a day per person. And this particular graph, that is only 5% of the amount of waste generated globally. The other percentages come from the larger waste streams, which are construction as well as agriculture. So there's a huge amount of these, what I term waste revenues out there that could be utilized for agriculture or crop production to meet these standards, to meet the needs for food production that we have. And there's also potential for these materials to have harmful human pathogens that could be put back into our system. So we need to look at these for their, to study them further to see if they can be broken down and rendered safe for use in production via vermicomposting. Excuse me. And so these are important things to look at. Just to bring it all home here in my conclusion, my research looked at vermicomposting and it showed that it could be a safe and effective way of removing harmful pathogens from manures, as well as these manures that were processed could meet FSMA microbial standards, making it compliant to be used in crop production, as well as pre-composting was not a necessary process prior to vermicomposting, which has many implications since globally not everyone has the means or the ability to get large equipment to process manures or other materials via thermophilic composting. And so many regions around the world have a form of epigeic worm species to utilize for composting. And so it could be a cost-effective alternative to help manage waste around the world. With that, I'd like to thank the University of Hawaiʻi Hilo for letting me use its facilities. I'd like to thank the TCBES program for accepting me, as well as my and teaching me the things I needed to know for developing my research. I'd like to thank my cohort for giving me guidance and also for being here today, as well as the analytical lab for processing my samples, and in particular Jasmine for helping me figure out the best protocol for pulling my samples to deliver to her, and then Haole Maloa Foundation for funding my entire research as well as my tuition. I really appreciate that. And I'd like to thank my committee for being there and being very helpful in guiding me and directing me throughout this whole process. And I'd like to shout out to my mom and my wife and my son. Thank you very much for being there and helping me out. Without further ado, is there any questions?
Questions from the committee or the general public? Thank you, by the way, for gracing or attending Jake's defense. Questions, please?
I have a question. Can you hear me?
Yes, I can hear you.
Okay. Can we go back to the results slide, the one with the bar graphs? Yeah,
This one?
yeah, right here. So for this one, it looks like the baseline is before treatment, before composting, before
Yes.
vermicomposting, right?
Yes.
So that's a very far left 2 bars.
Yes.
And then the third bar is hog manure. Is it after composting?
That's after composting, yes.
And how long was it composted?
So it was composted for 15 days. the average temperature from that particular pile was roughly 131 to 132 degrees Fahrenheit.
And how come the populations went up so much?
So in some of the research I was looking at, curing process, the process of letting the temperature drop and allowing for other organisms to come in, was a huge part at stabilizing it via general suppression methods, just by competition of other organisms. And also there was another article I looked at, which was specific methods of suppression. And they were shown that there's some species like Pseudomonas species that actually antagonize E. coli via antibiosis. So that could be one particular method. Curing was recommended for at least 6 months after the thermophilic stage drops off. But there's other articles showing that a minimum of a year was needed for it to be stabilized or to reduce the occurrence. So that's one potential reason I kind of inferred to deem why the increase of my hog manures E. coli and total coliform occurred.
Yeah, and I guess these bar graphs, those are error bars on it. How many samples did you process for each treatment?
So I had 40 samples in total, outside of the baseline samples, which were to determine the best dilution and if there was E. coli. So 40 samples, 10 from each particular, oh my God, each treatment.
Yeah.
And different manure.
So like five for each treatment then?
Yeah. To note too, my pre-composted, this was one sample per pile due to the cost of the rest of the total sample. So random numbers were generated off of a SAS for this one as well to equal 10 samples for statistical reasons.
Okay.
Yeah.
So it looks like the vermicompost, with or without the composting, was able to reduce the coliforms.
Yes.
Yeah.
Yeah.
So why do you think? What is the mechanism for that?
So my research didn't go into that, but I did a lot of research on it, and there was a couple articles that were looking at two, like I said, general and specific. So some were stating the with larger microbial mass in the finished compost, it just made competition for the harmful pathogens, and they would eventually not be able to compete with these particular more aggressive pathogen or aggressive microorganisms. Another mechanism for vermicomposting in particular, which was a little more specific, was by Dominguez, and he looked at material that passed through the gizzard of the worm species, Escherichia Andre, and he showed that when they inoculated material prior to feeding it to the epigeic worm, they took the material once it left the worm, once it excreted it, they took that and they sampled that, and they're noting that there was a process that happened when it passed through the gizzard of the worm species there. It changed composition and prevented it from E. coli's ability to occur after it was consumed. So it made it more difficult, I guess is what I'm saying, for it to survive through this passage of the alimentary canal.
Okay.
Yeah, if that answered your question.
Yeah. Good.
Hey Jake, I had a question.
Hey Marty.
Hey, so commercially, what I guess manure is used in I guess commercially, what manure of the manures that you mentioned in that one slide of the farm animals is used more frequently, or is used the most frequently?
I'd have to say it's for you're saying in specific for vermicomposting, right?
No, just in general.
In general, well, okay, I'll break it up into 2 points then. So, vermicomposting in particular, I'd say the heaviest used is cow manure, and in particular dairy manure. Not 100% sure why that is the particular one, other than that there's probably a potential for that there's a large amount of it. That and the facilities that usually have dairy cows, they use an expeller because they spray the floors down to clean the facility, and so they want to remove the liquids from the solids, and so they usually utilize a lagoon, and then they have all these solids that they have to get rid of. So since it's dropped its moisture content, it kind of makes it already an ideal composted or for hot composting. So most industries for vermicomposting pre-compost prior to vermicomposting for particular reasons, like if that particular facility utilized antibiotics or oh my god, worming agents to they don't want to harm their worms when they're doing the production of vermicompost. I'd have to say pretty much in the US in cities, one of the largest resources that is composted thermophilically is solid waste, human waste, after being from the treatment plants. That's the two examples for those two particular industries.
Okay, thanks.
You're welcome.
I have another question then.
Okay.
So, about how much stuff does an earthworm like go through? For instance, like, you know, in your pile of compost, you put a bunch of earthworms in there. Can you predict that like the earthworms will have like basically processed everything in that pile, or no?
So there's a general rule of thumb, and it is very general, that states that an earthworm can consume its body weight a day of the material. It's very general, but I got to go to a conference with Norman in North Carolina, and there is a very large vermicompost producer in Mexico that was there, and he talked about that is not the case. He had a client that was like, well, if you just bring me this many amount of worms, say 1 ton of worms to take this 1 ton pile, he's like, I'll bring it here and we'll see if that pile of worms, you know, breaks down and consumes that pile, which is not the case. Temperatures and other factors are never ideal inside in a vermiculture operation or a vermicomposting operation. I mean, there's the temperatures can fluctuate. The size of your worms can be different. The age of your particular worm species can be different. The material that you put in might not be a favorable material for them. There's a whole plethora of factors, but the general rule is that earthworms can consume their body weight a day.
What is a earthworm life cycle? How long from egg to egg, let's say?
It depends on the particular species. For Escherichia fetida, the worm has to be the earthworm has to be an adult, which means it has a collar, and that can occur from anywhere from 7 to 10 weeks after birthing. And then once they have a collar, cocoons can be formed regularly if there's an adequate population with older, also other mature adults that can breed. They're hermaphrodites, so they can use any worm that they come in contact with, they can reproduce with. Generally, I've seen maybe cocooning with mature adults within a week that they can produce a cocoon. These cocoons can contain 1 to 3 worms inside of it, depending on how healthy these particular cocoons and worms are. And also conditions have to be ideal for these cocoons to emerge, because they'll go into a dormancy if they don't have an ideal condition. So that can also extend your time for their lifespan.
Good. Okay, thanks.
You're welcome.
Any other questions? John, do you have any question?
You want me to ask my questions now, Norman? Or I can wait a little bit longer.
Yeah, we can wait a little bit. And any other questions from the general public before we let you go, and then we'll have a brief meeting with just the committee for Jake. Going once, going twice. All right, so if you can clap your hands, you may do so. Thank you, everybody, for attending Jake's defense. And Jake, great job. And we'd like you to stay a little bit longer for perhaps more questions, suggestions from your committee. So.
Oh, I can't leave yet, then? No, I'm just joking.
No, not yet. So everyone else, thank you so much for attending, and we look forward to seeing you in more defenses, I guess, from our master's thesis program. So for now, I'd like to let you go, and again, thank you so much for attending.
Thank you.
Love you, Jake.
I love you, baby.
I think there are 2 other. Ricky is still online.
Ricky's still online? He wants to hear the real questioning.
Or is he? I think I can remove him, yeah?
Yeah.
I think you have the ability to, yeah.
Okay.
Also, I think most of the ones online, they stopped recording for the committee.
Okay. Shall I stop the recording from here?
Are you going to make the recording public, or are you going to distribute the recording?
Part of it, they want to, well, it's up to Jake, but I think part of it will be made public by the TCBES program. I think they're going to put it on YouTube. So I think this part I can unrecord.
Yeah, you can stop.